Hello everyone, this is Ranamay Saha. I am a research scholar at Indian Institute of Technology, Kanpur, India. Let me share my screen first. So yeah, today at IEEE Census 2025, I will be presenting on electrochemical impedance-driven detection of viral RNA from human plasma. It will be comparative insights from PCR and RT-LAMP techniques. These are the gold standard nucleic acid amplification techniques, if some of you are aware of. Moving forward, uh, we'll be actually taking up hepatitis C infection because hepatitis C viral infection is actually a serious public health problem with an estimated 3% of the world population infected with HCV. 80% of HCV infections become chronic with time. The paradox is 19% of the people living with CHC are actually aware that they are having the infection and 30% of them are on treatment. As per WHO as well as NPG, HCV has to be eliminated by 2030. That is the good news, actually. So, looking at the current schemes, we have antibody screening, which actually cannot distinguish between a current or past infection, often provide false negative or false positive results. This is an expensive process and a labor-intensive process. Then we have LFT or liver function test, which is being carried out when already level 2 infection has started. So it is of no meaning and it actually is a delayed diagnostic process. Then we have qPCR tests, quantitative polymerase chain reaction tests, which are industry gold standard at current times. It is a confirmative test, but time consuming and no portability. It is again a quantitative test and detection happens in a couple of weeks after infection onset. So our proposed area of research is an electronic detection scheme for HCV RNA detection. This we have taken up because of current miniaturization of electronics. So EIS is very easily integrable with microelectronics. It is a simple, fast, and inexpensive technique. And in our case, we'll be utilizing gold nanoparticle labeling to improve the sensitivity of the entire detection scheme. So the proposed scheme is a quantifiable, so current or past infection is distinguishable. We will be doing a test rapidly, but still confirmatory test. Accuracy will be comparable to qPCR tests. It will be low cost, so no requirement of skilled labor. And we will be doing early detection as said before. So our objectives will be standardizing gold preparation and gold nanoparticle DNA conjugation protocols. Then we'll be doing RT-PCR and PCR experiments. We'll be designing and fabricating a microfluidic chip which can actually integrate dielectrophoretic concentration or DP concentration step and impedance measurements. So DP concentration and gold nanoparticle labeling, as mentioned before, will be integrated to improve the sensitivity and specificity of the device detection scheme. So development of RT-LAMP process will be the last step to make a simpler thermal cycle of only one step, and insights into impediometric detection of RT-LAMP and RT-PCR products will be carried out afterwards. Having said that, we'll be moving ahead to material and methodology part. The viral strain used will be an hepatitis C virus RNA provided by Institute of Liver and Biliary Sciences, New Delhi, India. The synthesized cDNA will be of 327 base pairs length. So on the screen, you can see the primers which has been designed and ordered from ILS India. The first set of primers here are for PCR primers, and then we have shown here the 6 sets of primers for RT-LAMP based EIS technique. So we have designed the RT-LAMP primers through taking up RNA sequence from NCBI uploaded in FIRST format, and then the design parameters are optimized using NEB LAMP primer design tool. And then as a step 3 or last step, we have taken up the primer set for HCV1A aligned in BioEdit 7.2 and modified for accordance with genotype 1, 3, and 6, which are prevalent in India. So the last step, a design of inner, outer, and loop primers generation. Okay, so one more thing is that the gold nanoparticle synthesis are actually performed through standard citrate reduction or Turkiewicz method, and gold DNA nanoparticle conjugation process has been carried out through Tetons protocol. As these are very standardized processes, I'm not going to the details of these steps. So then let's discuss about the chip fabrication process. Our microfluidic chip consists of 4 different layers. The bottom layer, one bottom layer IDE, which is fabricated using standard photolithography process with S1813 positive photoresist and a lift-off method. After that, we have an SiO2 insulation layer to separate 2 electrode layers. To mention here, our chip consists of 2 interdigitated electrode layers, the purpose of which will be discussed in the upcoming slides. So on the SiO2 insulation layer, we have the top layer IDE, which is again fabricated through the same process discussed earlier. And on the top layer is a microfluidic channel layer, which is fabricated in a 10:1 mixture of PDMS through replication molding process using a negative SO8 photoresist. As can be seen in the bottom right corner of the screen, the interdigitated electrode dimensions are shown here with a 40 micron of inter-electrode gap and a 55 micron of electrode width. The whole space electrode area is 10 by 10 millimeter squares. So the microfluidic channel can contain a total of around 100 nanoliters of fluid. Okay, so the bottom layer is an oxidized silicon substrate, which contains a 200 nanometer of oxide layer, and the IDs are of 30 nanometers of chromium and 120 nanometers of nickel electrodes, so making the whole chip a very inexpensive device. Moving ahead with the experimental setup, the schematic shown here actually represents the whole chip structure. So here, the schematic of the fabricated chip utilized for carrying out the impedance measurements is being shown. The basic device size is 25 plus 38 millimeter square in dimensions and consists of a microfluidic chamber made up of polydimethyl siloxane for PCR or RT-LAMP reactions and 2 interdigitated electrode layers for carrying out DEP confinement of particles. So DEP means dielectrophoretic confinement of particles, as discussed earlier, and impedance measurement. So these are the these are the purposes of using twin electrodes in our case. So the ID dimensions we have discussed in the earlier slide, and then the PCR product is passed into the microfluidic channel through the inlet, and we use a syringe pump of Harvard apparatus, and the fluids or the analytes are introduced into the microfluidic channel at a speed of 1 microliter per hour using a 5 micrometer needle. Moving ahead with the workflow, the overall workflow of the process consists of RNA isolation. Following RNA isolation, as can be seen in figure A3A here, PCR amplification of HCV is carried out using a thermocycler Eppendorf, whereas RT-LAMP amplification is carried out using a water bath maintained at 65 degrees centigrade. Both unlabeled and gold label primers are employed in each method. The reaction mixture is then introduced into the PDMS channel through a syringe pump and microneedle, as discussed earlier. A waveform generator is connected to the buried ID layer for DP confinement, and that happens for 1 minute near the electrode surface at an optimal frequency of 500 Hz and 50 kHz for unlabeled and gold label primers, respectively. Subsequently, the impedance data are recorded using an impedance analyzer connected to the top layer ID, as can be seen in figure 2D. So for DP characterization, the bottom electrodes are supplied with 10-volt peak-to-peak voltage at a frequency range of 50 Hz to 4 megahertz. The parallel capacitance value, which actually gives us the double layer capacitance, is measured through the impedance analyzer. And then the impedance data is recorded at the DP frequency, which gives maximum capacitance value. Then the corresponding impedance data is plotted and evaluated using an AC voltage excitation of 500 millivolt RMS over a frequency range of 20 Hz to 10 megahertz. The DP frequency is actually maintained for 1 to 2 minutes, and then the capacitance value is recorded after switching of the input supply to bottom electrodes. So this is done so that the signals don't interfere between the 2 electrode layers. So following that, we actually move to results and discussions. So here we have taken care of a confirmatory test for DNA nanoparticle conjugation using UV absorbance plots and transmission electron microscopy plots. So from the UV plot, it has been seen that the shift of absorbance peak from 520 nm to 523 and 524 nm for AuNP PCR primers and AuNP RT-LAMP primer mix actually confirm the AuNP primer conjugation. Secondly, that during transmission electron microscopy analysis, we have seen clumps forming up in when we have tested the conjugated solution. So the clumps actually indicate the conjugation process or confirms the conjugation process, as can be actually supported by literature also. So the concentration of the detected sample is 35 nanogram per microliter and 400 femtogram per microliter for PCR and RT-LAMP process respectively, as has been measured through UV absorbance at 260 nanometers. Regarding impedance measurements, impedance values are characterized by reducing arcs with increasing number of PCR cycles and further with gold labeling. The progressive drop in impedance with the increasing concentration or size of DNA is primarily attributed to the electrochemical changes in the solution. As the DNA has a negative charge, a counter-ion cloud is generated near the DNA backbone, contributing to the dipole moments. The ionic currents can be enhanced due to mobile counter-ions around the DNA. The citrate-capped gold nanoparticles further decrease the effective length, which in turn would result in higher charge transfer. So moving ahead, we can see the difference with PCR-IS, EIS, and RT-LAMP EIS. So the Nyquist plots of the PCR products exhibit a reverse trend in the two additional semicircles at higher frequencies, where the impedance response shows a modest increase as the PCR cycle number rises. This can be actually due to reducing surface charge transfer processes owing to shift of capacitive dominance over the impedance measurements to resistive nature. However, in case of RT-LAMP, the dense layer of long-looped hybridized DNA severely hinders the charge transfer right from the start, hereby actually opposing or suppressing the high-frequency response. The impedance behavior shifts to diffusion control regime at lower frequencies, giving rise to a Warburg component. The RT-LAMP EIS plots presented through Nyquist groups show a progressive reduction in impedance with increasing viral loads. A very low LOD of 400 femtogram per microliter has been obtained, which is comparable to the best results in literature and at least twice as good as the conventional RT-PCR fluorescence-based detection schemes. In contrast to preliminary RT-PCR EIS conclusions exhibiting two high-frequency semicircles, the equivalent circuit of RT-LAMP exhibits only one high-frequency semicircle due to its isothermal nature and simpler interfacial behavior. So the RT-LAMP products are longer and loop structures which instantly settle down over the electrode at very high frequencies, blocking any charge transfer processes. So these are the equivalent circuits which has been obtained through curve fitting of the plots. Moving ahead, if we look at the effect of dielectrophoretic confinement, we can see in case of labeled and non-labeled PCR products as well as RT-LAMP products, it can be directly observed that the impedance value substantially reduces after DP concentration of the analyte in both cases. The plots march to some common points as the frequency increases in both the cases, because as frequency increases, the impedance moves mostly towards the resistance side. So here we can see that the main reason for reducing arcs or reducing impedance values with DP confinement is DNA accumulation in the regions of high electric field intensity, leading to positive DEP effect. And then the effect is more pronounced in case of RT-LAMP, as can be observed here. Again, enhancement of DEP effect with ANP labeling can be observed as nanoparticles also undergo positive DEP. So regarding the conclusions, impedance spectroscopy has been utilized as a tool to electronically detect polymerase chain reaction products of hepatitis C virus, cDNA and reverse transcription loop mediated isothermal amplification products of HCV RNA samples. TEM images in UV spectroscopy results are used as confirmatory steps for DNA nanoparticle conjugation. Metallic labeling and DP-based concentration of analyte are included for improving the sensitivity and specificity of the electronic detection scheme. The developed method achieves a detection limit of 35 nanogram per microliter and 400 femtogram per microliter for RT-PCR and RT-LAMP respectively, and the process completes in 3 hours in PCR and 80 minutes in RT-LAMP, while the sensing process begins within 15 minutes of sample introduction, supporting its potential use in rapid and portable diagnostic applications. These are the references.